Good morning to Dublin on the Green Island. Congratulations to the organizers for BlooDHIT. And thank you for inviting me to this first session of this inaugural conference of a new series of transfusion medicine meetings. Let it become a hit. I'm speaking virtually to you in an attempt to leave a greener footprint. And I will be online for a discussion after my presentation. My topic is on opportunities in red cell genotyping and genomics research. Genotyping and genomics research. I do have no conflicts of interest. I like to point out that a couple of those essays discussed do not necessarily have approval in all jurisdictions, and you have to make sure that you are in agreement with the regulations in your jurisdictions. With appropriate validations, all these essays can be used in any country. I'm a US government employee, but I'm speaking for myself here. So it is very appropriate that this inaugural session has two major topics, because in a recent State of the Science in Transfusion Medicine Symposium organized by the NIH here in the US, two major improvements over the last 30 years in transfusion medicine were identified as infectious disease testing, with the last major advance with hepatitis C in the early 1990s, and molecular typing for blood groups, with most major advancement occurring since the mid-1990s and more so in the 2000s and ongoing. Now, these two major improvements have very many similarities, but also they're different. Infectious disease testing obviously is super important for safety and mostly concerned with donor testing. Recently, with some pathogen reduction technology for platelets and plasma added, and notably, there is little that we can do for red blood cells when it comes to pathogen reduction, which, however, represents obviously the majority of our products. In difference to that, molecular typing for blood groups is important for safety, and I'll show you data for that in a second, but also important and has a major future impact on effectiveness of our transfusion medicine therapy. It's done with donors, that's very much increasing in the moment, and also with patients, a little bit lacking behind because of the logistics. And there is some matching done by molecular methods. This component is also increasing. Now, hemolytic transfusion reactions were always number two in the reasons for fatalities caused by transfusion. This shows data from the US from more than 10 years ago, where infectious diseases were clearly lacking behind the hemolytic transfusion reactions. And this is shown here with microbial infections, not limited to viruses. And I'm sure you're familiar with this report here from a current report from SHOT, showing data from Great Britain, where there is one hemolytic transfusion reaction causing a fatality, but no transfusion-transmitted infections. So we are making improvements, but hemolytic transfusion reactions are still out there, and you can have a lot of morbidity, even if you don't die, caused by hemolytic transfusion reaction, but there is no morbidity if you don't get an infection transmitted by transfusion. So these are the distinctions between the two fields, and they were identified as the major fields of progress. Now, talking about molecular typing for blood groups, which I call red cell genotyping, one could ask questions: what is an example for the implementation of blood center? How do the numbers compare with red cell genotyping relative to the time-proven serology, and what is its impact on servicing clinical needs? Now, almost 10 years ago, we published this study showing the integration of red cell genotyping into the blood supply chain. The data are from the blood center of Wisconsin, and we compared 30 years of experience with serology with the then newly introduced genotyping. And as you can see here, in 30 years, a little more than 72,000 donors were tested by serology, and within 4 years, 43,000 donors were tested by genotyping. The minimum number of antigens tested per donor in serology was 1, and the maximum for very few donors was 28. This compares to genotyping, where the absolute minimum number of antigens tested was 26. And more typically, 42 antigens were tested. And the limit here was the size of the platform. Today, more antigens can be tested. So summarizing these data, we could show that more than 300,000 antigens were tested over 30 years by serology, compared to 1.6 million antigens tested within 4 years by genotyping. And this is, if you look to these data, this is close to revolutionizing our field, because serology managed to test a little bit more than 10,000 per year over decades, whereas genotyping could provide data of more than 400,000 per year on a very short, over a very short period of time. And there is little doubt, although no cost calculation was done, that this testing is cheaper, certainly per antigen, but also overall. Once this genotyping was implemented, serologic testing for those antigens was stopped. And this clearly is based on the cost efficiency rationale. But here's the real impact. We showed that the genotyped red cell units supplied 95% of all clinical requests. And the reason that 5% could not be serviced is because these antigens were at that time not on the chip for 2 reasons. For one of them, the molecular basis was not known. That has changed. Now, these days, all antigens could be tested by molecular means. And the other thing was that there was a certain limit to the platform, so one could not test more than 42 antigens. This has also changed. Now, if one wants to accomplish that today, 100% of clinical requests could be fulfilled. This shows data from 2013 over the year in a sliding window of 7 collection days, and the availability of red cells, which was roughly around 500 throughout the year, and 30% of them were available with genotyping data. Now, there's some attrition in donors, so one has to continue testing donors to keep that, which actually was done in the past 10 years. And it's certainly possible to reach a higher rate than 30% if that was needed for clinical purposes. At that point, people often ask, what about costs? And well, it's important to consider costs, but I don't quite understand why this aspect is considered as so critical for molecular immunohematology, when if there's a new emerging virus, a lot of money is put at addressing the viruses, despite the fact that the evidence over the last 30 years shows that there is more risk to the health and the life of the recipients caused by blood groups and mismatches than by actual virus threats. And maybe we have to think about these aspects in a different way to capture the opportunities for the benefit of our patients. If we are too restrictive on the red cell genotyping, just because of the costs, this may not be in the best interest of our patients. There are also hidden costs of alloimmunization. Here is a very recent study from last month showing the median costs per inpatient discharge, comparing patients in dark blue without alloimmunization and in red with alloimmunization. This is adjusted for DRGs. There may still be confounding involved, but there is accumulating evidence that it's better to have no alloantibody than to have an alloantibody. Also, when it comes to the safety of your transfusion supply. Now, let me address quickly what is the right technical approach today. There is rapid testing with typical turnaround of hours. This can be done for single patients. The buzzwords here are SSP or Sanger sequencing, and this approach will be needed for a while. It's available in particular in Europe since around 2005, and it will continue be needed for cost and turnaround logistics aspects. But clearly, the move is going to massive parallel sequence. This has a little longer turnaround, not considering the logistics. And the real benefit is if you can test many patients in batches, such as with next generation sequencing. To accomplish this, however, this is typically done by computerized prediction of the alleles based on reference sequences. And I want to go with the next slides in explaining how we should create databases for reference sequences. So why do we need them? Now, the computerized allele prediction is somewhat error prone. That's the reason why we base it on reference sequences, which is widely applied in and successfully applied in HLA using next generation sequencing, abbreviated as NGS. The frequencies of such reference sequences in populations is known. So it can, the frequencies can be used for analyzing the NGS data. They may also differ in populations, and you can use that when you know the ethnicity of the given patient. So there are databases for, it would be good to have databases for reference sequences, in particular when they are very accurate, because they are needed as templates for phasing the NGS data. With these techniques, you get the information from the 2 chromosomes as one genotype, and then you somehow have to split that ideally into the 2 alleles, one from each chromosome. And this is facilitated by reference sequences, and it's called the phasing of the data. The utility of that is highly demonstrated in clinical HLA typing. They have an excellent database for the assignment of HLA types. No such database exists for blood group genes, and one of the reasons is the lack of reference sequences. So where do we get the reference sequences from in our field, which are required for establishing databases? There are 3 ways, and I show you examples how to do that. It can be through sequencing of population cohorts, typically done in a combination of 2 NGS technologies to avoid errors. The NGS technologies do have known errors, but the type of errors differs between the technologies. So if you mix and match the right NGS technologies, you can actually control one of those technologies with the other. The second approach is extract the data from genome databases. This requires bioinformatics algorithms, and I'll show you an example. You also can predict alleles that have not been observed by phylogeny, and this is an approach trying to determine ancestral sequences, so the precursors of the currently observed alleles. The known sequences must share some common precursor. And the ancestral sequence of this common precursor can be calculated, and the likelihood can be determined. And this number can be used also for clinical diagnostic, or at least improving the clinical diagnostic. Here's the first method, the thorough sequencing of populations. I would call that a hard way. So we did that, many people did that. Here's an example where we did it for 21,000 nucleotides in the region of the ERMAP gene, which is the standard blood group. We sequenced 50 individuals, and we did find 72 SNFs. So sequence single nucleotide variants. And they can be associated with each other in various ways. And one would have to match them, to phase them, to get the right sequence. So the theoretical possible number of matching them is 2 to the power of 72. So no one can handle that number. The actual number, once we resolved it by molecular physical testing, was only 48. So these sequences were identified and verified. They can be considered experimentally confirmed, which means error-free, and they can be used for computerized allele prediction of NGS data. Another way is extracted from genome databases. I would call that the convenient way. You get the reference sequences from genome databases, such as the 1000 Genomes project with bioinformatic algorithms. And if you use the right algorithm, then they also can be considered experimentally confirmed and error-free. And this is how it's done. We actually found in the Duffy gene, when we extracted them from the 1000 Genomes project, 902 distinct sequences. And if you look here to this little speck at the bottom of this slide, which looks like dirt, this is actually a sequence, the worst sequence that we got, which after all has 1,900 nucleotides, which is way more than you need to make a Duffy determination by confidence. So if you do Sanger sequencing, you typically get something like 200 or 300 nucleotide suites. If you do it with SSP test kits, you're lucky if you have 10 nucleotides to make a still reliable determination of the Duffy phenotype with confidence. So here we got in the worst case scenario 1,900 nucleotides, and then of course there were 900 more sequences, in particular up here, with lengths ranging from 40,000 to all the way up to 80,000 nucleotides. Now one could ask the question, why do you need 80,000 nucleotides if 10 nucleotides are enough to do that with confidence? Well, the quality of your results get much better if you have longer sequences, and we should not complain about a lot of data if we want to move our field into the field of big data. We should actually cherish this approach. Now, the third way is predict by phylogeny. I would call that the smart way. And this is to analyze the evolutionary relationship of sequences and predict intermediate sequences. And this is the way how it's done. We analyzed our 48 ERMAP alleles that I showed you in some previous slides. We analyzed the phylogeny using standard Bayesian analysis. It's a standard approach research typically done for viruses. So we actually collaborated with an individual who did that for HIV. And this was in 2019, so that was before COVID, but since then the exact same approach has been applied for COVID virus as well. And the idea is to predict unobserved sequences at so-called internal nodes. This is shown here. These nodes here called I, K, J, and L were actually observed, and therefore the sequence is known. So that's not news. However, it is also possible to predict the other nodes here labeled A to H. They are not known, they are rather predicted, and now the catch here is that we actually can calculate the so-called posterior probability, which is a technical term, but it gives you a mark how likely the occurrence of such alleles is. And we calculated all possible ancestral sequences at each internal node. The result was to get 4,243 predicted sequences. And this compares very favorably to the theoretically possible 2 to the power of 72 sequences. You can deal with 4,200 predicted sequences, and it's possible to order those sequences according to their probability and use it for clinical prediction. The utility is that they can be used to along with the confirmed sequences, and both sets then combined improve the accuracy of phasing sequences, considering that once in a while you will run into a new allele that you haven't seen before, and the set of known alleles doesn't really help you, but the set of predicted possible likely alleles may help you quite a bit in figuring out what the accurate sequence is. And this is shown here in this set of algorithms. I walk you through it. You start out with the set of known alleles from the genome databases or from thorough sequencing of population. Those alleles are experimentally verified and can be used for the analysis. You can also then do the phylogenetic analysis that I just showed you using the Bayesian approach. And in the end, you have a set of known alleles plus the predicted alleles. At the same time, you do next generation sequencing and you get with high throughput technologies, and then you combine these two to actually infer the alleles that your individual has. And then there are 3 possibilities. The easy one is that you actually find that the individual has 2 known alleles, and that's actually also the most common result, which is straightforward for your clinical report, and there is still an error, rate of error, but it's low. You may then also find that perhaps one known allele is present and one of the potentially ancestral alleles occurs. This occurs with a low frequency. The conclusion should be restrained in your clinical report because there is some risk of error. And if you have time, then you wait for the experimental confirmation, which should be done in such cases. And you have an indication that this is a candidate for further in-depth analysis. The third possibility is that there is no known and experimentally verified allele involved, and it would be 2 possible alleles. That's of course has a risk of error. You're dealing essentially with 2 unknown alleles. How should you know what the right answer is. This then clearly would also go into experimental confirmation to resolve that issue. And we should have a conveyor belt approach that these analysis is actually continued either in your own lab or as a send out, which is probably the better idea, to a national reference center, but it should be followed up. And obviously this would go around and around, and at every round it would get better and better and better, and the databases would improve, provided that this is done in a somewhat non-competitive approach with a national resource. So what's next? We need more donor red cell genotyping, preferably high throughput, massive parallel next generation sequencing. This should be relatively inexpensive, and blood centers need to be properly equipped. This is in full swing, but not nearly fully implemented. There's still a lot of improvements possible even in Western healthcare systems. There is clearly a need for patient red cell genotyping, and one of that will be addressed later today in a different session at your BlooDHIT meeting, because there are patient cohorts who benefit more than others, or who's more needy than others, and we should actually help them by preferentially addressing their needs through molecular typing. If you can't do that in your own hospital, then a logistics should be established to refer the samples to get a reasonable turnaround time. Now, getting back to this NHLBI State of the Science in Transfusion Medicine symposium of earlier this year, the mechanistic aspects of components were addressed at one topic, and saying we need to approach them with the omics testing to characterize components, or to characterize the donors, which then would also characterize the donated blood from those donors. Red cell genotyping is one of these omics approaches called Genomics. I like to point out that whatever we establish for red cell genotyping and blood groups can equally apply to any other genetic variant that may be of relevance, and it's quite likely that this is not limited to blood groups at all. In the end, it's a question of terminology. Why do we call a gene a blood group and others not? At the molecular level, the approach is exactly the same. So all what I showed could be applied to other genes, and we are not the only ones working in the field. So in the end, it will be a collaborative approach. The other aspect, which I'm not addressing here today, is mass spectroscopy protein analysis called proteinomics. This also has a lot of potential, also has more variability, while the genes don't vary that much at all. The proteins in a blood bag can change a lot, for example, during storage of the red cell unit. Both aspects will massively contribute to the safety, as I said at the beginning, but more so, and this is a very important take-home message for today, it will contribute to the effectiveness, the quality of our transfusion outcomes, such as better oxygen delivery, therapeutics delivery, and other red cell functions. Much of that still needs to be explored, but this is clearly something that has a lot of potential. We just found the tip of the iceberg of this approach, and there is a lot that can be derived, in particular if we don't shy back from the big data approach, but rather cherish it, that we can have such enormous databases to work with and figure out what is causing and contributing to improving the effectiveness of our therapy. New methods in transfusion medicine were summarized, in particular data science, with bioinformatics, biostatistics, big data that can be used for multifactorial analysis. This is a highly competitive field. So to bring people with that knowledge to our field requires that we respect them, because you probably cannot pay the sums that other industry may be able to pay for such approaches. So we should very carefully develop this field, which essentially will then apply artificial intelligence and also implement machine learning. The phylogeny approach, for example, could be run as a machine learning approach if one had the tools to apply that. There's an intricate path from genetic variant to clinical implementation. Very many fields are working on that. This here is a review from almost 10 years ago. And I don't want to walk you through this complex system, but I want to make a final statement here. It's not going to be easy. Be patient with the people working in this field. Tolerate the costs. We need to invest money in the interest of the benefit of our patients, accept pragmatic approaches. So don't ask for 100% foolproof technology before you would even touch this. And I'm saying this out of the experience with molecular blood groups, where I often heard that unless this technology is 100% safe and comparable to serology, we wouldn't even touch it. That may not be in the best interest of our patient, because incremental improvements are better than no improvement at all. There is a long-standing trend in transfusion medicine, and while I spent as physician in transfusion medicine in the last 37 years, I saw that happening all the way from whole blood to the very refined blood products that we have today, by trying to remove non-essential blood constituents from the therapeutically necessary blood constituents. So leukocytes and plasmas, for example, is removed. If you want to transfuse red cells, we cannot remove the red cells, but we can make sure that the potentially harmful constituents, such as antigens, are not present on the therapeutically required red cells. If you do that on an individual, personalized medicine approach, honoring what a patient needs or can tolerate. So in the end, this will be a blood product recipient dry matching approach, so blood matching by genotype only, without serologic testing. Some people call that an electronic match. Coming back to my initial slide, there were these two major improvements over the last 30 years. These are probably also the two major opportunities for the next 30 years, although I would like to flip the order. So I would argue that red cell genotyping for blood groups, but also for other genetic variants, so for the non-blood groups, is important for safety and will gain enormous importance for effectiveness of our therapy. And dry matching at the genetic level can be the major advancement, as it is clearly a determinant of transfusion outcome beyond safety. Infectious disease testing, just to make that statement, is of course critical, and it's necessary to remain alert for rapid testing of emerging agents. But I want to make also a statement as a perhaps discussion point. We should also be proactive to implement pathogen reduction technologies, in particular for red cells, even if these technologies are not super efficient. A little reduction of risk can go a long way, in particular when it comes to the emergent agents, and these two advancements combined will determine a lot of our research activities and improvements for the benefit of our patients and their safety. Thank you.
Thank you very much. And do we have a connection to Bethesda?
We do. Hello.
Dr. Flegel, good morning. It must be pretty early for you.
Well, it's 6 o'clock in the morning. Yeah.
We're just waiting an audio connection, unless we're muted.
All right.
Can we hear you? Can you hear us?
Yes, hello, I hear you. Yeah. Do you hear me?
Yes, please go ahead. And thanks again for such an amazing talk. How are things in Bethesda?
All fine. So, too bad that I couldn't come. Yeah. I'm happy to discuss if there are questions. Yeah.
Do we have any questions from the audience? Professor Madrigal.
Good morning over there. Good morning over there. Hello. Professor Madrigal, myself. It's fantastic talk, and what you're talking is really fascinating and sometimes difficult to follow. But given the fact that there is so much information, as you say, on the HLA distribution in population and the sorts of progression in terms of differentiation of the different alleles, can you correlate your blood groups or what you are doing in addition next to the large number of people? I mean, there are 42 million people typed for HLA in the world for unrelated bone marrow donors. So would you be able, there is a lot of material available, in particular in DCI-BMTR, would you be able to correlate in linkage in a way with your genomic analysis of the blood groups?
If genomic DNA is available, then it can be used to run for the testing for other genes, such as blood groups. That would be possible, and one could tap into that resource. Would be a feasible approach, which allows also to then determine the population frequencies, as the HLA samples are typically associated with the ethnicity. That's the first approach with the cohort testing, population testing in cohorts. That's what I addressed. This can be done, yes.
Because obviously the introduction of next generation sequencing in the, as you say, with multi-allelic typing on HLA has actually allowed to have a very large information.
Yeah.
So probably the data must, source of relevant will be the one in Seattle from Effie Peters, or I will come back to you later on this. Very interesting.
Perfect, thank you. Mm-hmm.
Any final questions? Just for a headline for us, how far away do you think we are from dry matching in the laboratory for patients? Or should we see that first from the NIH even?
With manual technology, it's already done. So it is implemented to some extent in some institution, and it's clearly increasing. We would need better computer tools to facilitate that and make life easier. It's way too cumbersome and time-consuming in the moment, but it is done. So we are applying that here and more so at blood centers who have many more donors and units.
Thank you very much, and thank you for your talk. We'll see you at the American Society of Hematology, hopefully.
All right. Thank you.
Bye-bye.
Bye-bye.